Hello, everyone. Thank you for joining us today for another one of the very compelling talks that has been organized by Professor Lars Frelund and his committee at the International Society for Vaccines. Today, we have the privilege of having Professor Teressa Lambe, who's the Karolinska Head of Vaccine Immunology and Professor of Vaccinology and Immunology. She's also a Pandemic Sciences Institute investigator at the University of Oxford. Professor Lambe has been really crucial for developing the Jenner Institute and then AstraZeneca COVID vaccine, for which she received the honorary OBE from the United Kingdom and was also awarded the Distinguished Service Award in Ireland. So she's going to speak to us today about a number of how to approach outbreak pathogens and develop vaccines against them. So I'm going to turn it over to her so we can have the maximum amount of time. If you have questions, you can type them into the Q&A at any time point. And then also, if at the end of this, then she will answer questions. And some of you, if you prefer, we may also just unmute you to ask your question. But if you type them in, then we'll know who has questions and who to address at the end. So, Professor Lambe?
Thank you so much for the very kind introduction. I hope you can both hear and see my slides well. I'm delighted to be invited here today to talk about vaccines against outbreak pathogens. What have we learned and what's next? And I like to start a lot of my presentations with a very old slide that I've used for many, many years now. But it really brings it home to me how important vaccines are. To my mind, vaccines are one of the most important medicinal interventions we have in our arsenal of effectiveness against infectious disease. But it also reminds me that when I started out in this field, the types of characteristics I was looking for in a vaccine are not necessarily the characteristics that are front and foremost in my mind when I'm developing a vaccine now. And this is a very old infographic from the Public Health England in collaboration with the World Health Organization, or WHO, which demonstrates the impact that vaccines can have. This is the number of diseases within the UK before a vaccine was introduced and then after a vaccine was introduced. So what are the types of characteristics that I now obsess about when I'm thinking about making vaccines? Well, first and foremost, we need to make sure that vaccines are tolerable so that they induce side effects that are minimal and will result in compliance, and that individuals will see a whole immunization schedule through. You ideally want protection, you absolutely want protection against severe disease. Ideally, you would also like protection against asymptomatic disease, onward transmission, and less impactful disease. But really, you can't always achieve this with the vaccines. But at a minimum, you want protection against severe disease. And ideally, you would like that to be sustained or long-lived. And to achieve that, you need to induce an effective immune response. So you need to induce both antibodies and T cells effectively and prime them, and also lay down the maintenance so that you've got long-lived memory so that you are protected against recurring disease exposure. The aspects that I certainly did not consider when I was developing vaccines at the very beginning of my career were the practical considerations, the price point or the cost. And truthfully, this is probably the main driver now, because I've come to realize that if you're making vaccines against diseases of importance in the global south or low and middle income countries, you really need to make them at a price point that they will get into the arms of the people you're trying to protect. So it's great having a brilliant idea, but if you can't actually manufacture the vaccine and get them to the people who need them, your great idea will not, unfortunately, amount to a lot when it comes to sustaining life. And one of the impacts that will feed back into the costs is the stability of your vaccine. So how long a shelf life it has, because the longer the shelf life, the lower the cost. And also the other aspect that I consider is delivery. How are you going to get your vaccine to the people and into the people? So do you need extensive cold chain storage, or do you need less refrigeration? Because again, that will drive the cost point down. Also, are you going to give a vaccine in the arm, or are you going to give a vaccine up the nose? Because again, that will impact the manufacturer and will impact the cost. And I like to end this slide with this cartoon of Edward Jenner's time. And Edward Jenner is frequently referred to as the grandfather of vaccination. And that's a little misleading considering vaccination, virulation, or controlled exposure to infectious disease to protect an individual had been going on for many centuries before Edward Jenner. But I digress slightly. This physician noticed that milkmaids who had been exposed to cowpox were then not susceptible to smallpox. And smallpox was an absolutely devastating disease. It was thought to have claimed in the region of between 300 and 500 million individuals. So Edward Jenner performed some very rudimentary clinical trials, the type of clinical trials that we would not do today. He exposed individuals to pustules from a milkmaid who had cowpox at the time, and then challenged them with smallpox, and they were protected. So this was a huge step forward in the field. You, in essence, could get protected against smallpox through virulation or inoculation. But even back then, in the late 1700s, there was a sentiment, a vaccine hesitancy, or even an anti-vax sentiment. And this is a cartoon displaying that sentiment, suggesting that you would go on to develop bovine-like characteristics if you were to get Edward Jenner's vaccine. Nobody has turned into a cow because of vaccination, but it really speaks to the theme of vaccine hesitancy, vaccine reluctance, and anti-vax sentiment that was around in 1700s and remains with us today. And it's definitely something that we do need to consider seriously when we are generating vaccines. We need to build trust so that we can get vaccines into the arms of people who need to be protected against disease. So in 2013 to 2016, there was an outbreak of Ebola virus disease in Western Africa. And this outbreak was thought to have claimed some 11,000 lives. It's thought to have been a vast underestimate of the number of fatalities because of that outbreak. But really, after this outbreak, the WHO and many other very eminent bodies came together and generated a hit list of those pathogens that they thought might cause the next outbreak, the next pandemic or epidemic. And some of these viruses are very well known. So I suspect everybody on this call will have heard of Ebola virus, probably Zika virus. Crimean Congo hemorrhagic fever doesn't roll off the tongue, but nevertheless, it was identified as a pathogen of concern. And we would, in the University of Oxford, with many, many other entities and collaborators globally, started to really push the dial and have a concerted effort to develop vaccines against these outbreak pathogens. So how do you do that? How do you develop a vaccine against an unknown or an outbreak pathogen? And hopefully in this next slide, I'll be able to explain it to you such that you can have my job tomorrow and I can retire. But in the first instance, if there is an outbreak, the first thing you have to do is identify the pathogen that is causing the outbreak. And once you've identified the pathogen that is causing the outbreak, you then use that information to identify targets as part of that pathogen that you think if you had a strong immune response against, you would be protected against overt disease. So for SARS-CoV-2 and for most of the coronaviruses, and truthfully, many, many viral targets, it's that cell entry factor that we need to target. It's the part of the pathogen that facilitates the virus getting into your body and causing disease. And if you can stop the virus entering via neutralizing antibodies or other, then you're going to protect an individual from overt disease. And in essence, that's what the darlings of COVID did. The viral vector technology and the lipid nanoparticle messenger RNA technology used that information. They took the genetic code, or the recipe, if you will, for the spike protein, and they carried that information into your body so that when you were vaccinated, your body started to make copious amounts of the spike protein of SARS-CoV-2. Your antibodies and your T cells did not recognize this unknown entity and mounted a strong immune response against it. And in essence, that immune response, memory T cells, neutralizing antibodies, were ready and waiting for when you became exposed to SARS-CoV-2. So in some ways, you were training your body for when it would be exposed to COVID-19. So one of the questions I frequently get asked is, how could you go so fast? What was it that you did differently? And truthfully, we didn't do anything differently. We just compressed it. This is the typical development process and timeline for a therapeutic or a drug, and it can take anywhere between 5 to 20 years, truthfully. And you start small, you start with your vaccine design. And myself and a colleague designed the Oxford AstraZeneca vaccine over a weekend. But truthfully, this vaccine design can take months, if not longer, if you've got a complex organism or a target that it's difficult to identify which part of you need an immune response against to temper infection. But once you've designed your vaccine, you then progress into preclinical studies. Again, you start small and you typically start in small animal models. Once you've demonstrated that you are getting an immune response in these small animals, you may progress onto larger animals, such as NHPs or other. And you're looking really there, if you can, to do a challenge experiment to demonstrate that your vaccine, when used to immunize NHPs, can protect against the disease it's been designed against. After the preclinical studies, you will start to make vaccine that is of quality that can go into humans. And each of these steps, as you progress along this development timeline, increases in time. So as I've said, the vaccine design can be done over a weekend. Phase 1 GMP takes months, if not a year, depending on the complexity of what you're manufacturing. But really the step change that happens as you progress along is the amount of investment that is required. It's not a step change, it's astronomical. Apologies, I'm now getting messages. So really what you need to do is ensure that your vaccine is the best in class as you progress along this timeline. So it is an iterative process. You will end up at times taking a step backwards to make sure you've got the best in class vaccine that you're going to progress on to phase 3 GMP and onward licensure. And that was the type of development pathway that we were looking at when we started to make a vaccine against a different coronavirus. Middle East respiratory syndrome is caused by the MERS coronavirus. So it is a coronavirus like SARS-CoV-2. It has a lower transmissibility and its case fatality is higher. So you've got a case fatality of in and around 34%. It generally impacts individuals that have got comorbidities, and frequently those individuals that are working in close proximity to the zoonotic reservoir, which is the camel. So we did what we typically do. We took the information about the coronavirus. We looked to see which target we should include in a vaccine, and it was the MERS spike protein. We took that genetic sequence and we put it into our viral vector. And then we started our animal studies. And as I've described, we started small. We were able to demonstrate small vaccine efficacy in small preclinical models. And we were also able to demonstrate efficacy in NHPs. So a single dose of this vaccine was able to protect monkeys against pneumonia caused by MERS-CoV challenge. And that gave us the confidence, and our funders the confidence, to facilitate a GMP manufacture. And we're very fortunate that we have an on-site manufacturing facility at the University of Oxford where we can make vaccine that is suitable to go into clinical trial. So we initiated that GMP manufacture, and then we started our first in-human trial in Oxford in March 2018. We enrolled 24 individuals, and they all received a single intramuscular vaccination. we were able to demonstrate that the vaccine was well tolerated. So typically what you will see with these types of vaccines is you will get pain at the injection site, and you will frequently have side effects that are similar to a common cold or flu-like illness. And when we mapped the immune response against Chatter-Swan MERS, we were able to demonstrate that we were getting both antibodies and T cells. So really, we had this information that we could generate a vaccine that was at least immunogenic in humans and protective in animals against a related coronavirus prior to SARS-CoV-2 coming on the scene. But once SARS-CoV-2 did come onto the scene, once we realized it was a coronavirus, we looked at the development process and timeline, and we realized we could not do it in the typical manner, in the kind of sequential manner. So instead of doing it sequentially, we stacked it. So we did all the normal steps you would do, and it goes without saying that the first thought that we had in our minds was the safety of our volunteers. When you were doing clinical trials, that's your first, middle, and last thought when you're delivering those trials, is to deliver them safely. So the risk that we took in delivering the trials in this manner was a financial risk. Because before we knew that we had a vaccine that was immunogenic or protective in animals, we had started our GMP process. And by the time we had started our phase 1, we had started our phase 3 GMP. And as I've already explained, the amount of money that is needed to progress these types of stages is astronomical compared to the vaccine design stage. So we didn't know that we would have a vaccine that would be protective, but we were hopeful because of the promising data we had on a related coronavirus. So we started in this manner and were able to develop a vaccine and test it in less than a year. So as I've already alluded to, we started small. The team was, I think it was 3 to 4 people at the very beginning. This was me making up the first batch of vaccine that was going to go into animals. The sequence was released on the 12th of January 2020, and 35 days later, by the 26th of February, we knew from our small animal studies that we had a vaccine that was immunogenic. But really the animal work then started in earnest and the team started to grow. And we worked with absolutely fabulous collaborators ranging from Australia to the US and the UK. And we tested the vaccine in a lot of animal models. This is not typical of what you would do when you're developing a vaccine against an outbreak pathogen. You don't tend to do as many animals models as we did in 2020. But if you remember back to 2020, there was at that stage a worry, a concern that a vaccine against a coronavirus might induce ADE, antibody-dependent enhancement. And ADE has been seen in humans with certain diseases and had also been seen in some limited animal models with certain vaccine types for other coronaviruses, principally SARS-CoV-1. And in essence, what ADE is, in individuals or animals that were vaccinated against a pathogen, when they were then exposed to that pathogen, they actually developed worse symptoms. So instead of being protected, they actually had a worse outcome in challenge. So we did a lot of work with hamsters, guinea pigs, transmission studies, and full pigs and ferrets to really look and see if we were seeing any hint of ADE, and we weren't. And ultimately, it was some fabulous work with great collaborators in the Rocky Mountain labs that demonstrated that 1 shot or 2 shots of our vaccine were protective in rhesus macaques against SARS-CoV-2 mediated pneumonia. And as I've already alluded to, while these studies were ongoing, we had a GMP manufacturer in our small on-site facility, CBF, run by the fabulous Kat Green. So the first doses were made in April, and we were ready for our clinical trials by the 22nd of April. And by the end of April, we had an agreement to partner with AZ. And again, AZ were absolutely fabulous partners throughout this whole journey. And at that stage, there was over 2 million cases worldwide. So our phase 1/2 clinical trials started. We were oversubscribed for the clinical trials, and we had some absolutely wonderful volunteers who didn't even need to roll up their sleeves. They came prepared to get immunized. I think one of the lessons that I have learned through that journey was the impact of social media and negative messaging, and in some cases, active misinformation being put out there. And for this lovely volunteer, I think it was also a learning curve, because she woke up the day after, or maybe it was 3 days after she got immunized, to a fake news story that she had died. And we did a lot to try and disarm the misinformation. But truthfully, once that genie is out of the bottle, it's really hard to get that back in. So it's an ongoing struggle that we have around vaccine communication and active vaccine misinformation. But it's not something that we shrink away from. I think there is an emphasis on scientists to communicate effectively and simply so that we can distill the correct information and honest information. I'm not in any way advocating that individuals should enter into dialogue with active vaccine misinformation, but it is something as scientists we do need to consider as part of our jobs, in my opinion. So as these trials were ongoing, we were ramping up our phase 1 clinical delivery, and we were really getting into the nuts and bolts of looking at the immune response. And that's when we started our phase 2/3 clinical trial delivery. And at that time in the UK was doing what it should have been. It was in a lockdown, so we were unable to test the efficacy or if our vaccine could protect against SARS-CoV-2. So while delivering our phase 2 and 3, we reached out to our wonderful collaborators globally. So Suan in Brazil, George and Mayanga in Kenya, and Shabir in South Africa. And through this wonderful partnership, we were able to demonstrate that our vaccine was protective. So the steps and the lessons that I've taken away from this journey, first and foremost, is plan for the future and plan many steps ahead. Plan A, B, C, or D may not be what you get to implement, but if you keep on making small steps forward, you will get there. I absolutely think that you need to partner with pharma early and partner with an organization that has the same vision and the same goals as you do. And we were very lucky in the work that we did with AstraZeneca, Serum Institute, and many others. You do need to work at risk, but the priority is always the safety of the volunteers. And as I've emphasized, the risk that allowed us to develop and others develop vaccines in less than a year is a financial one. And collaboration is absolutely key. And you need that global collaboration across industry, regs, funders, NHS, healthcare providers, and others if you're really going to turn the dial on manufacturing a vaccine of this type of impact. And I think it would be fair to say that one of the things that we as a huge global team are most proud of is that more than 3 billion doses of this vaccine have been released for supply in over 180 countries. And most importantly to us, approximately 2/3 of those doses went to low and lower middle income countries via COVAX. And it was estimated that over 6 million lives were saved in 2021 alone. And I think this is definitely something that the team are hugely proud of. But what next? What do we do with the learnings that we've amassed from SARS-CoV-2 to better prepare us? And if you look at some of the other outbreak pathogens, such as the filovirus family, Ebola virus, or Zaire Ebola virus, if you've been working on it for as long as I have, was discovered in 1976. And it took 15 years, truthfully probably longer, to get a vaccine from the first publication of there being a vaccine all the way through to licensure. So how do we do better? So we need to look at this drug and vaccine pipeline, this 10 to 20 years, and really identify those bottlenecks and see if we can turn the dial. Because truthfully, some of those vaccines that we have developed against outbreak pathogens are wonderful and marvelous and can and should be used, absolutely. But they don't always hit the target product profile that has been suggested as what we should emulate or aim for some of these diseases. So at the moment, we have vaccines that are licensed that are protective only against one strain of filovirus, where really you would like a vaccine that is protective against multiple filoviruses concurrently. I'm not the only one that has had lessons learned through SARS-CoV-2, and really it was some of the lessons that came out of a meeting with the G7 leaders where they welcomed the 100-day missions that I think will turn the dial and result in a paradigm shift. So the 100-day mission, it's almost a moonshot. It's the ability to really design, develop, test, and get vaccines in arms in 100 days from when a public health emergency of international concern or similar is announced. So how can we do that? How are we going to be in the best position to really turn the dial and achieve that type of mission? So this was a report that was authored by scientific, government, and industry experts. It had a number of recommendations to enable readiness of both diagnostics, therapeutics, and vaccines for the next pandemic. And the aspect that interests me most readily, although it's a really interesting read, is the commendation to produce a library of prototype vaccines. So really, if you think of the SARS-CoV-2 pandemic, it was that information that we had around MERS coronavirus that helped us think we could make a vaccine of impact against SARS-CoV-2. But in a lot of cases, we're starting here. We're starting at a stage where we have no research already performed on the virus or the viral family, and we don't have data in phase 1. So what about turning the dial and actually really amassing data in the interpandemic period that helps identify the best way forward for developing vaccines against viral families? So amassing data across research and phase 1 delivery so that if a pandemic were to strike, we would be in a much better position. So for us to be able to do that, we really need to know the culprits that we're going to try and combat. And there have been a number of these activities spearheaded by the WHO, CEPI, the UK Vaccine Network, and many others, NIAID, etc., where they have looked at the world's greatest pathogen threats and they have categorized them such that we have now got a list of families that we think may cause a pandemic, epidemic, or public health emergency. So what about amassing information around these viral families, looking at different platforms of vaccines, looking at different antigen design, so that we could help inform if it was that pathogen, but equally if it was a related pathogen. And that's some of the work that the team are doing with support from CEPI. And working in collaboration with Moderna to use their RNA technology and also our viral vector technology. And the viral family that we are targeting is New World arenaviruses, and we're targeting Hunan virus. It's an arenavirus that causes Argentinian hemorrhagic fever. There is a vaccine licensed in Argentina against this virus. It is a live attenuated vaccine strain, and it has been identified that it only requires 2 amino acid changes for that vaccine strain to revert from being non-pathogenic to pathogenic. So we're trying to both develop a vaccine that may be better suited, but also to amass a large body, a playbook, if you will, of information against New World arenaviruses, such that if there was an outbreak, we would be in a position that we could go faster and hopefully aim for that 100-day moonshot. So we're working with some absolutely fabulous collaborators, a team in the UK, a team in Germany, and a team in the US that are looking at the antigen and using different ways to both stabilize it, increase expression, and augment presentation. And I think the simplest way for me to explain this, and I'm not a structural biologist, is that they are using a huge array of approaches to achieve this type of antigen stabilization and technology. Some of those are what I would refer to as informed antigen design. So looking at what has worked before on related viruses. Some of it is using AI and machine learning. And ultimately, what we have done is we have achieved 12 different types of antigens that have been stabilized to a different degree. And we are putting those antigens in both an RNA technology and a viral vector technology. And we are progressing it through that development pipeline up to phase 1 that I've already described. So we are looking at the immune response in small animals, and we are looking at the efficacy in guinea pigs. And then we will go on to down select our best candidates to progress onto human clinical trials. So this is the work that we are doing with a number of brilliant collaborators and teams to delineate which antigenic approaches are best for New World arena viruses to develop vaccines of impact. So in essence, if we look at the lessons that I have taken away from SARS-CoV-2, how are we addressing them? So first and foremost, planning for the future and planning many steps ahead. We are now generating preclinical and clinical data during the interpandemic period. And this is only one of many programs that are being pushed forward to help generate that playbook against a number of viral families. I believe you need to partner with pharma early. So we're partnering to combine our academic expertise. And I would have to say that academics, in my opinion, and I'm slightly biased, we are great at blue skies thinking. We are great at rapidly escalating and pivoting. We don't have, in general, and this is a gross generalization, we don't have that level of manpower or woman power to really get these types of therapeutics all the way through to licensure, uphold the license, and implement rollout globally. So working with scaled programmable platforms such as that seen with the RNA technologies and working in partnership with Moderna has been really fruitful. We are working at risk. We are exploring prototype pathogens and viral families in parallel. And again, that's to generate that playbook that will put us in a better position going forward. One of the other great aspects about being a academic is that you can use innovative immunology to help de-risk and progress programs. So what do I mean about that? I mean that in some of these studies, we will absolutely deliver what is needed to identify those types of antigens or those vaccines that should be progressed onto clinical trials. But as we are doing that, we can also address some of the questions that are outstanding in the field around priming an immune response, around looking at the longevity of an immune response. And we also will frequently do work that will look at what cells in the lymph nodes are using fine needle aspirates to really delve into the immunology of how these vaccines are working, so we can discern what is good and what is bad and what we need to target. Also looking at mucosal delivery and really delineating how the mucosa and the systemic intramuscular delivery, how they differ and what the overlaps are. I obviously can't count, this should be number 4, but collaboration is key. And maybe it's been ranked number 1 for a reason, because truthfully, without that global, industrial, academic, and policy alignment, you're not going to be able to progress these types of programs in a manner that can make an impact globally. And I think it's important to focus on the global network that both we have built up and more probably to discuss the global network that CEPI and many other partners, WHO, etc., have built up. Because I firmly believe that if you're going to impact human health, large-scale scientific collaboration and programs are needed. You can't be expert on everything, and I certainly am not expert on everything, but I know who to reach out to if I need help with different aspects of that development pipeline. You need inclusive, not exclusive practices. All of these different parts, and in fact, I've left policy off, which I shouldn't have. All of these parts are what are needed to develop these types of playbooks to really turn the dial and better prepare us for the next pandemic. We're also pretty invested in doing the standard immune testing, but twinned with investigative immunology. So to really do a deep dive and look at the type of immunology results that we can get out of these types of studies. So what are the typical types of questions that we will be able to address with these studies? We do a lot of system serology work. So this is system serology work that we've done in collaboration on SARS-CoV-2. This is an influenza vaccine that we've looked at both at the antibody responses across many different types of assays. We do a lot of flow cytometry. I used to get very excited when we could do flow cytometry of 8 or 12 colors. We now have collaborations that enable us to do flow cytometry in-house with 57 colors in clinical trials. We do fine needle aspirates followed by single cell RNA-seq and bulk-seq to really look at those cells that are driving the immune response. And I think it's important to also consider different ethnicities when we're doing these studies and also different age groups. We work with partners who have developed tonsil organoid models so we can look at the mechanism of action of these vaccines and partners, again, just across the road that have tissue on a chip type of technologies that allow us to, again, look at the types of cells and how they are interacting. And again, most importantly, are those assays that are done routinely, day in, day out, that are standardized, qualified, validated, that will really get your vaccine progressed onward to licensure. So we don't de-invest in this type of technology. And I should also make the point that viral vectors and RNA vaccines are not the only vaccine technologies out there. And I don't think we should only invest in that type of technology for preparedness for the next pandemic. I think we need to invest in a wide gambit of different types of technologies, such as protein and adjuvant, inactivated, VLPs, etc. So we really need that whole gambit of vaccine technologies to best prepare us going forward. So in this manner, we, and along with many other people, are generating a playbook for optimal antigen design, assessed in multiple vaccine platforms. We are generating, just within my lab, vaccines against outbreak pathogens across a number of different viral families. So there's a lot of people working very hard here. We want to progress from bench to bedside. We don't just use vaccines to figure out the immune response. We make vaccines so that we can protect people. But while we're doing that, we will use experimental medicine, investigative immunology, and a systems biology approach to determine correlates of protection, those mechanisms of action. How do you prime a good immune response? How do you keep it maintained? And really deconvoluting those mucosal and systemic responses. So it just remains for me to thank everybody who's been involved in this whole journey that covers my whole career, in fact, in vaccine development. But first and foremost, thanking those clinical trial volunteers and many, many other teams that helped develop the Oxford AstraZeneca vaccine. This is my team as of today, and this is a picture of the Oxford Vaccine Group where we sit. And we recently had our 30th year celebration. We've been in existence for that long. And to recognize some of the wonderful partners that we have. And truthfully, I will likely have forgotten some. So I will end my talk there. And very much looking forward to any questions you might have.
Thank you very much for that superb talk. We have a number of questions, and I'm going to start with Dr. Michael Francis from the International Veterinary Vaccine Network, and I'm going to actually unmute you, Michael, so you can ask your question directly if you would like to do that. And then we will move to some of the typed in questions as well, but people feel free to either raise your hand or to type in your questions.
Thanks, Margaret. Can you hear me? Yeah.
Yes.
Yeah, good. Hi, Teressa. Thanks for that. Teressa, do you think that the use of the ChAdOx1 as a vector for multiple human pathogens may become a potential problem in the future? And is this likely to result in some immunological competition within the host?
Yeah, that's a good question. So I think your first part is asking around anti-vector immunity and whether we can reuse the platform. And correct me if that's not correct. So what we have seen is we've done some work where we have taken the ChAdOx1 SARS-CoV-2 vaccine and we have administered it in repeated doses. And certainly from what we've seen up to third dose, you're still getting an immune response being augmented against the spike protein. We've also done some studies where we have taken individuals that got a different ChAdOx. So I think it was some of the individuals that got ChAdOx1 MERS, and then we vaccinated those with the ChAdOx nCoV-19 or AZD1222. And actually for naive individuals and those individuals who got the ChAdOx1 MERS, again, we see an antibody response that is of a similar power. And that's not to say that I don't think that there may be some impact of repeated if you were to use it almost 6 months, but I also think the interval between that repeated use will also impact its usefulness. So you can certainly with viral vectors reuse them. These are other viral vectors supporting data to what I just said. You can reuse them after 6 months, 1 year, 12 months, and see an augmented immune response. I don't know if that addresses the question in completeness, Michael.
Yeah, I think it does, but I mean, you're creating quite a long list now of different antigens within ChAdOx, and I just wondered how people might prioritize the use of those in the future.
Oh yeah, that's fair. And I should truthfully say we're using different platforms. We're not targeting all of these with ChAdOx. We're targeting some, but not all, with different technologies as well. And I think if you were to look at where Ebola virus outbreaks are occurring and where Hunan virus occurs, they are different geographies, but you're absolutely right. You can and do have multiple outbreaks of concern in the same geography. So looking at alternate technologies as well. I'm definitely somebody who thinks more is better. So having multiple types of platforms, I think, is the way forward as well, Michael.
Thank you.
Right. Thank you. So we have some chat questions. The first one is, thanks for your presentation so far. I noticed an emphasis almost exclusively on viral pathogens. Do we need to worry about outbreak bacterial pathogens? And if so, would the strategy be the same?
Yeah, that's a question I frequently get where I work, and I answer it with a slightly, I'm a simple person. I like to work on simple pathogens. So I do predominantly focus on viruses. I do have, I'm being quite, I do have partnerships and people working within the same institute working on bacterial pathogens. And no, you're absolutely right. The same types of technology won't always work for pathogens that are more complex, such as bacteria. Although there are some really interesting data coming out for plague using the types of technology. So.
The next question is, I read a paper in Nature Medicine recently saying that B-cell memory does not happen effectively with mRNA vaccines for SARS-CoV-19. As such, we needed to go for several boosters with COVID-19 vaccine. Do you think the mRNA technology needs to be improved? Thanks.
That's an interesting question, and I'll start with I was frequently asked during the pandemic, what vaccine should I get? And I know that's not what this question is, but I would truthfully say get whatever vaccine that is offered to you that will save your life. So I don't work on making RNA technology more stable or modifying it. But I do know that there are individuals who are doing that. And truthfully, I think we should invest in this type of investigations with all the technologies. If you consider some of the data that came out around the heterologous prime boost type of work, you can see absolutely amazing immunogenicity that seems to be longer lived and maintained. So for me, as an immunologist and vaccinologist, what is it that the immune system is doing that allows that absolutely amazing priming and really good maintenance? And how do we ultimately make that happen in ideally a one-dose vaccine? So that's something that I'm definitely very interested in and avidly pursuing.
Yeah, just as by way of background, what was interesting about that particular paper was it specifically was looking at saying that the SARS-CoV-2 plasma cells weren't durably
bone marrow.
established in the long-lived compartment. But it looks like there may be some issue about the virus itself and the antigen, the spike protein, so that I think that it may be more complex than just mRNA, although clearly the antigen for with an mRNA vaccine may not persist as long as other types of vaccines, some, notably, of course, live virus vaccines and so on. So I think it's a really interesting question, but also potentially more complicated in addition to any issues about mRNA vaccines. So yeah, thanks for that question. The next question is actually from Matt Price, and he says, how might you approach epidemic or outbreak pathogens that tend to strike fast and burn out quickly, often within your 100 days, and yet still carry a very serious public health or societal burden, such as Ebola?
Yeah, that's a good question. And it's one that I know the WHO also think about very avidly. So one approach, and I'm segueing slightly, because for me, when these types of outbreaks happen, you want to get in, you want to help protect, and you want to, if you can, get vaccines in arms as rapidly as possible. And you don't, because these outbreaks will typically burn out. And I am touching wood, as an old Irish saying, to hopefully the next pandemic or epidemic also burns out quickly. So the WHO are looking at an iterative clinical trial type approach where you accumulate data over many outbreaks, working with partners in these countries where the outbreaks happen, so that you can accumulate your data across a number of outbreaks to help vaccines get licensure. And I think what you have definitely seen with the outbreak that happened in Rwanda, where the vaccines got into arms really quickly, it can be done. So it's not, while I do agree that they can burn out quickly, it's often that concern around what if they don't burn out quickly, that we really need to have vaccines and that playbook ready for it.
I think, though, that this raises another interesting issue, which is that, for example, there was Lyme vaccine that was licensed and was being sold many years ago. And what happened was actually, in addition to a lot of misinformation that got spread about it, people actually lost interest in it because in fact, it wasn't that Lyme went away. It was just that it wasn't as much of an issue and not on the front page news, and the numbers weren't as great. And so I think that this question actually relates even to a different aspect, which is that that unless it's something that people are really concerned about at the moment, then there are problems of policy, sustainability, frankly, even from a marketing perspective, because if it's something that's losing money, then there's not really an incentive for the companies to make it. So I think this is really an interesting but complex question, as well as this whole issue of how do you test and develop something within that 100 days. I think it's broader than that even.
No, I would agree completely with that. And truthfully, I think we're all a little bit tired and jaded from SARS-CoV-2 and talking about the pandemic. And I'm sure some of that talk was traumatizing for many people on this talk. And in some ways, if we now speak to government or other agencies around trying to amass funds to allow these types to get behind manufacturers, there's a lack of interest because it's almost it's old news and it was done before, so why do we need to invest in it now? And I think we need to change the narrative. We need to almost make it less about an insurance policy and more about bolstering GDP in your own country, about that ecosystem that you're establishing, and the kind of investment in life sciences will result in your economy growing. And the type of innovation that you can and will have will feed into many different sectors. So that's certainly one way that I try and have those types of conversations now.
Yeah. So we have a question from Ben van der Zijs, who is asking specifically about adeno-based vaccines. So they seem to work well. The only serious problem is the TTS, which is the small but real incidence of thrombosis with thrombocytopenic syndrome. Can that be solved?
Yeah, it's a really great question. I thank you for asking it, actually. TTS, as you've already said, is very rare, but very serious. There have been steps in being able to diagnose and treat it more rapidly and readily, and certainly the incidence of the disease seems to be different in different geographies. So there seems to be a higher occurrence in Northern Europe as compared to other global geographies. As to whether that is genetics or geography, that has not been deconvoluted. There is work going on around looking at modifying, well, truthfully looking to see what the real mechanism of action of TTS is. How do these adenoviral vectors, how are they associated with TTS? And once we've got that mechanism of action, and there is a supposition around PF4 antibodies and autoantibodies there, but once we've got that truly delineated, we can look at modifying viral vectors. But we need the right animal models to really dig into this in the first instance. And I know there are some great teams working on those animal models. So I think there will be progress made. But I think it may be slower than I would like, but I'm somebody who would like it tomorrow, truthfully.
I think that this gets at the question that gets at some of the really fundamental issues for vaccines, which is that the benefit is obviously in the favor of benefit versus risk for the vast majority of people. However, for those small numbers of people, this is clearly not an outcome that you want. And I think that what's nice about your answer is that we as scientists and vaccine people need to focus on, getting to the facts and understand things as quickly as possible. It's not simply to say, well, the risk-benefit ratio is all in the favor of the benefit, because if you're one of those small number of individuals, clearly that's a terrible outcome, and I think we need to be very positive about it. I actually wanted to raise a question about One Health, because your presentation focused on humans, and yet clearly for many diseases, as for example, the avian flu that's all in the news now because of spread to mammals and then between mammals, this becomes an issue. So how do we best address this? Because it isn't simply thinking about protecting humans from animal diseases. It's actually that we're all part of one ecosystem, and we need to think about protecting the animals as well, for their own sake, but also because, for example, many flu vaccines are made in eggs. So if you have to kill all the chicken flocks, you can't make those vaccines, et cetera. But it's all basically very intertwined as we're all part of the same ecosystem. So could you just address what is being done to address the One Health aspects, but also how we as scientists can better address that?
Yeah, no, 100%. You've pretty much summed up a different talk I give around how it has to be a One Health, zoonotic focus as well. And I would say that I have an honorary position at the Pirbright Institute, which focuses on animal health. And a number of those vaccines, so Rift Valley fever is a vaccine of huge importance in animals. A colleague of mine, George Wareing, and somebody who's working within the lab, is working on vaccines to be delivered not just to humans, but also animals. I think when you are developing veterinary vaccines, you really need to consider the price point, because they need to be almost pennies, and you need to consider the market as well. So it's not that it may be more difficult to convince people who are in that field to vaccinate their flocks or their animals if they don't see an added benefit to maintaining their livestock. So if the animals do not succumb to the infection, take for example CCHF, but they pass that infection on to humans, there has to be a benefit to those individuals to vaccinate against CCHF. And one way to consider doing that is to consider a multivalent vaccine. And in doing that, you will also push the price point down of those vaccines. So it is absolutely something that I do think about, and the team thinks about, because there's no one person who can do this on their own, in my opinion.
One last comment, if I make, is I think that you mentioned this, but I think that besides looking at the different diseases, which is always challenging because you sort of have to be able to look in the future and you don't know which ones will arise, which ones will cause epidemics or pandemics, but is the focus on technology. So all of the vaccines that you mentioned for COVID were actually injected. And there was one vaccine developed by CanSino that was a mucosal vaccine. But I think that is a key issue that we need to focus on as well, which is the technologies, because the more infection we can prevent also means that the fewer new variants will arise because they're not spreading to as many people and so on. So could you just, in our last couple of minutes here, make a comment about other technologies, including mucosal delivery, for example?
Yeah, completely. And in fact, I would have said what you have said. So mucosal delivery is absolutely critical, in my opinion, for looking at preventing transmission as well, not just infection, but transmission. There is a supposition and data to support that the nasal immune response is not as long-lived as some other compartments. I think we need to figure out why. I also think there's a lot of work there that is needed to be done around the manufacturing of vaccines such that they can actually be delivered effectively in different parts of your respiratory system, so both nasally, aerosol, and in your lungs. I would also extend out, Margaret, to intradermal delivery, and intradermal delivery not just via needles but also patches, and how all of those three can actually put us in a better place for the next pandemic. The viruses that worry me, almost the ones that worry me the most, are always respiratory viruses. And that could be because I started working on flu, but it's certainly the one that I always revert back to. So the type of work that I think we need to look at for the mucosa is, how do we prime a good immune response? How do we maintain a good immune response in the upper respiratory? And how do we manufacture vaccines and devices such that they're at a price point that we're going to be able to stop those types of infections that are not more expensive than the tech that's out there already?
Okay, thanks. We had one last question that came in, and I want to thank you also for the intradermal delivery, because in fact, is a WHO approved device that was used in India for an authorized DNA vaccine that was in the skin and didn't therefore contribute to the 14 billion needles that have now been added to medical waste for COVID vaccines alone. So Victor Aswani has said, thank you. And what are the state of vaccines against arboviruses, which are causing epidemics more frequently? And we have 1 minute left for your answer.
I will say with Victor, I think we need to do more work on arboviruses. It's not my field. I've got enough viruses, but I know many brilliant people working on it, so I wouldn't worry about them too much.
Okay.
Although I do worry about them.
Good. Thank you so much, Professor Lambe. This was great. And thank you to the audience for your fantastic questions and your participation. Please join ISV and please participate in all of the future programs that we have. So thanks to everybody for joining us today. Bye-bye.
Thank you for hosting me. All the best. Bye-bye.
Yeah.